Hello everybody. Now we come to the last lecture in module 5, um, building a multiple sequence alignment, in session 2 using GAL view. So in this one we'll learn how to select and edit sequences using GAL view. So it very simply we can do selecting and editing sequences in GAL view. So it's as you wish to. For example, arbitrary, you want to select some region, random region, or you would like to select a complete column or complete sequence. You just press on what you want to select, and then you can copy and edit later. So what you will do is just put your mouse or click on your selection, and you just copy it, and then after you copy you just go and paste it into a new alignment. So you can select a part of your big alignment, and this small part which you are interested in, you can copy it in another file and then put it into the alignment. So you simply, as you will see now, you just you take, for example, if you would like to take a small random region, you just in the top, you just press to the top left corner, and press and hold, and then drag the mouse until you come to the right corner, the bottom right corner. So first you put your mouse on the top left corner, the place you would like to start the dragging, and then go down until you come to the bottom right corner. Then your selection will be you can right click on that, and then you can copy it and paste it in another alignment. So this is a possibility in GAL view to select a small region. Another possibility is to select, for example, if you put your mouse over a residue and you would like to select all the residues in this sequence, so you just put your mouse over a sequence, a residue, and then drag to the down clock, just clicking the first position and then hold down and press shift and click the other one. This will give you a range of position. So if you would like to have a range of position in the same column, for example, or the same in the same sequence, so you just press the first one and hold shift key, and then press the last one you would like to put it in the range, and then it will keep this range will be copied. If you would like to have a discontinuous region, so you just press Control, put your click on the first position you would like to add, and then and keep pressing Control, and then try to select others using also clicking on the other position that you want to select. So simply what I want, simply you can either select continuous region hold by holding shift, or discontinuous regions by holding control. And I think this the same techniques can be used in Microsoft Word, for example, to the same two keys can be used in Microsoft Word to hold to have a continuous selection or discontinuous selection. Anyway, of course, if you would like to have to select complete sequences, you just click down onto the ID in the left hand panel. You can just select whatever sequence you want to select, and you can use the same techniques using Control for discontinuous, and using Shift for continuous selection. Of course, you always can change between your cursor mode and mouse mode using F2, as we mentioned before. And if you would like to mark in a cursor mode, if you would like to use cursor mode and have a selection, so in the top left of the selection, you just press Q. This will mark the top corner, and in the bottom corner you just press M. Of course, after you having a selection or selected part, you can simply make inverted selection. This is very useful feature, very useful tool. You can because sometime you would like to hide the other part than your selection. So you can select a region which you are, for example, a motif or active site or whatever in your protein alignment. You can select it. After that, you can say invert selection. Invert selection means that the program will select everything except what you have selected initially. So, and then you can hide these inverted selection regions. So this means that whatever will appear is only what you have selected initially. So this is a very good technique to really show only what you want to show. So you have two options actually in this case. Either you just select the region you want to select and then copy it in a new window, or if you don't want to do that, you just select this and then invert selection to select everything except your initial region, and then you say you press on hide selection. So in this case, you will hide everything except your what you have selected. Like this, for example, after I select whatever my part would like to the part that I'm interested in, and then I just in the view menu, I just say press on hide selected region. So in this case, he will hide selected region. But if I right click on this selected region, I will find the option to hide everything except all but selected region. So he will hide everything except this selected region. And of course, I can show this again, as we mentioned before. We can show this hidden region again. This first, as we said, this is a very nice thing. We can group this selected region, this selection. We can right click. If you right click in selection, you can put this group of sequences, put a name or characteristics for that group selection. So if you have a conserved region that you are interested in, you just can highlight them and then right click, and then you say selection and then edit group name. For example, you put a name for that part. If you're interested for that, you can say extremities, active site, whatever thing you are interested in, as we mentioned yesterday. So you can put a name for this group of sequences and put that name here. So you can know that next time when you see this group of sequences, you know what they are. And if you want to publish this part only, you can copy that part and then as you in a new window, and then you can after coloring them and do whatever work you want to do on them, you can just print them in image to have them in your publication, only the part that you are interested in. This also you can save them to use them in other programs. Like if you have a part that is a domain, or you would like you cut this part you're interested in and you want to use other program to analyze this part and do more analysis to that part. So for example, you can copy them and then save them in any output you want, like pair, cluster, W, FASTA format, MSF, PFAM, and then save them in that different format, which can be used in other program to extract more information from that sequence. Anyway, this is the last lecture. JalView can give you a lot of lots of possibilities for analysis and of multiple sequence alignment. And the good thing about JalView that you can use it with other programs. You can import data from different multiple aligners, and you can expand and export data from JalView to different aligners also. So this kind of exchanging data and information between JalView and other program make it really very powerful tool for multiple sequence alignment. I hope this session was informative for you, and hope to listen to all your queries and answer all your questions. And of course, hope to see you again in other courses, and enjoy the list of the sessions and lectures in the other modules. And have a good time, and goodbye.